Okay, so mitosis. Now, at first, I just want to highlight some of the main points of each subphase of mitosis. If we call mitosis a phase, well, it can be divided into 5 subphases or stages. So a cell leaves G2, enters mitosis, and those 5 stages of mitosis are as follows. We have prophase, which is followed by prometaphase. Then comes metaphase, then anaphase, and then telophase. Now I'll diagram each of these individually, but with respect to prophase, the chromosomes condense, the nuclear membrane breaks down, and these things called kinetochores assemble on the centromeres. During prometaphase, the mitotic spindle forms. An enzyme called separase degrades a protein complex called cohesin, which exists between the sister chromatids, and it degrades these cohesin complexes all along the sister chromatids except at the centromeres. And then the chromosomes are moved to the metaphase plate. Now the metaphase plate is an imaginary line that sort of travels down the middle of the nucleus. So metaphase is my favorite because it's, the stage when the chromosomes are aligned on the metaphase plate. During anaphase, there's a compound called sugoshin, which is degraded. That allows centromeric cohesin to be degraded. That allows the sister chromatids to separate. And the sisters are pulled to opposite poles. During telophase, the chromosomes arrive at the opposite poles. The microtubules, which were pulling the chromosomes, disassemble. Nuclear membranes form between the two groups, around the two groups of chromosomes, and then the chromosomes decondense. Now, after this, cytokinesis can happen. Now, many textbooks sort of separate cytokinesis, which is division of the cell or division of the cytoplasm. They treat it separately from mitosis, and that's what we do in this course. We will leave cytokinesis, division of the cytoplasm, division of the cell, which occurs after mitosis. We'll leave that to cell biology, what is cell biology? BSc 203. Okay, now let's take a look at each of these stages in more detail. Okay, so ideally you are taking notes as you are watching this video, and you have a nice list of all of those things that happen during the various stages of mitosis. So you can refer to that list as I'm going through the various stages. So let's take prophase first. Now, instead of considering a human cell that has 46 chromosomes, I will, consider a make-believe diploid cell with 4 chromosomes. And, okay, let's go with 2 metacentric chromosomes and 2 acrocentric chromosomes, and these are 2 pairs of homologous chromosomes. So in this cell right here, now mitosis is about, taking one nucleus and making it into two genetically identical nuclei. So at the very beginning of prophase, this nucleus right here, what's going to happen? The DNA is going to condense, and we're going to be able to see the chromosomes. If we were to stain that cell that is in mitotic prophase, we could see the chromosomes appearing because they're condensing, and we can stain them and we can see them under a microscope because we can't really see them when they're not condensed. So there's one of our condensed chromosomes here. Here's another one over here. And notice how I've diagrammed them as sister chromatids, chromosomes containing sister chromatids, because remember, this is mitosis. This is after G2, which is after DNA replication. So all the chromosomes should be diagrammed as pairs of sister chromatids. So, okay, the chromosomes condense. The nuclear membrane breaks down during prophase. So let's see. I wonder if I can quickly erase the nuclear membrane. Let's see. Ah, it did. It worked. Okay, great. Nuclear membrane breaks down. And the third key thing that happens is kinetochores form on the centromere. So we're going to get one kinetochore forming on either side of the centromere. So what is a kinetochore? Kinetochore. So the kinetochore is where these things called microtubules will attach. The microtubules attach to the centromeric regions, but they don't attach directly to the DNA. They attach to proteins that form the kinetochore on the centromeres. So the kinetochore is the protein complex that attaches to the centromere, and microtubules will attach to the kinetochores of each chromosome. Okay, so that's prophase. Now let's take a look at prometaphase. I think I can use this same diagram here. So prometaphase, what's happening there? So the mitotic spindle forms. Now, outside of where the nuclear membrane was, once that's gone, just outside, on opposite sides of the nucleus, there's going to be a region where we will find centrosomes. So two centrosomes. Now the centrosomes contain these organelles called centrioles. Now these are what form the microtubules. So the microtubules will grow from the centrosomes. I won't refer to the centrioles. I'm just going to call these the centrosomes. The microtubules will grow or extend from the centrosomes to each kinetochore. Oop. Okay, so these are called microtubules. They're like little tubes. And they're made of proteins called tubulins, alpha and beta tubulin, usually. So that's an extra detail we don't need, but we need to know these are microtubules and they extend from the centrosomes to the kinetochores of the chromosomes. So each centrosome is going to be connected to each of the kinetochores. Now, once those connect, okay, what else is happening? Well, the chromosomes are going to be pushed and pulled to the metaphase plate. And the metaphase plate is going to be this imaginary line that goes down wherever the nucleus was, where the nuclear membrane was. If we can draw a line down the center of that nucleus, well, that's the metaphase plate. And that's where the chromosomes are going to end up in a line during metaphase. And they're going to be pushed or pulled there by the microtubules. Now, also during prometaphase, we're going to have something else going on. So each pair of sister chromatids the sister chromatids are held together by a complex called cohesin. Now I'm conceptually diagramming it like this, but in reality, when DNA is replicated, let's say this is a strand of DNA, and during S phase that is replicated, so now there are two strands of DNA. Now cohesin is a complex, protein complex, that holds these two strands, close together. And so cohesin is found throughout these sister chromatids. And so these two arms, chromosome arms, are going to be very tightly associated during prophase. But the cohesin gets degraded by an enzyme called separase. During prometaphase, and it only gets degraded along the arms. So there's also cohesin holding the sister chromatids together at the centromere, but that cohesin is not degraded during prometaphase. Why? Something interesting happens. So that cohesin is coated with another protein called shugoshin, which I believe is a Japanese term for guardian spirit. So during prometaphase, the cohesin at the centromere is not degraded by the enzyme separase. But everywhere else along the chromosome, the cohesin is degraded between the sister chromatids. So, okay, that leads us to metaphase. Now let me start with a new diagram for metaphase. So now I can show those sister chromatids arms being really floppy now, because they're no longer held together by cohesin, except at the centromere. Now, during mitosis, the chromosomes are going to be lined up one on top of the other. And the reason I say that, and I want to emphasize that, is because when we talk about meiosis, the next class, there are two metaphases. In the first metaphase, the chromosomes, homologous chromosomes, line up side by side, not one on top of the other. But during mitosis, all the chromosomes line up, we can say vertically, on the metaphase plate. So we shouldn't see any chromosomes, any centromeres to the right or to the left of the metaphase plate. They should all be lined up one on top of the other. And now we can see this nice spindle shape of the microtubules attached to the centrosomes. and the kinetochores, which I won't diagram on the chromosomes. And this is metaphase. Now, remember, cohesin is holding together the sister chromatids at the centromeres. And that holds the sister chromatids together and holds that centromere as a single centromere, keeps it from breaking apart. Now, after metaphase, at the beginning of anaphase, Shugoshin is degraded by another enzyme. I'm not sure the name of that enzyme, but the enzyme is produced at the end of metaphase, and it finds that Shugoshin and it degrades it. And then the separase now can find that cohesin, centromere cohesin, and it can degrade it. Once that happens, the microtubules contract and the centromeres split, and the chromatids, individual sister chromatids, start moving towards opposite poles. So let's call this one pole. We'll call this the other pole. And so after the cohesin has been degraded at the centromere, microtubules contract, and the sisters are pulled towards opposite poles. So how I can diagram that like this. And microtubules. Oh, I lost one chromosome, didn't I? Okay, so how many chromosomes do we see here? We see 8 chromosomes now, right? Before we only had 4, but now we see 8. But 4 are moving that way, and 4 are moving that way. So that is a hard concept to understand, right? And it's difficult because, you know, the amount of DNA didn't change. So it's just by convention how we define numbers of chromosomes. By convention, we define it by the number of centromeres. So in this diagram right here, we see 4 centromeres. Down here, after the centromeres split, okay, now we see 2 centromeres, where before we only had 1. So now we can see 8 chromosomes here, but we have 4 moving towards each pole. So that's anaphase. And finally, telophase. Okay, what happens during telophase? The chromosomes arrive at each pole. And maybe I can just copy and paste this guy right here. What else happens? Well, the nuclear membrane, well, the microtubules disassemble. The nuclear membranes form. Now these can decondense, these chromosomes can decondense because, right, the cell's about to enter interphase, right? And as we said before, cytokinesis can now happen. Cytokinesis. Kinesis, say whichever one you want. Division of the cytoplasm. Again, we are going to consider cytokinesis to be totally distinct from mitosis, and it happens after mitosis. But this nucleus over here is going to be the nucleus for one of the cells resulting after cytokinesis, and this one over here is going to be the nucleus for the other cell resulting from cytokinesis. Okay, so that's mitosis. Again, so my plan this semester is to hopefully have you guys watch these videos before class, and then we can talk about some problems and anything that you're confused about during our class in-person class sessions. Okay, see you during class.